Welcome to the Performance Around the Clock podcast. I am Professor Satchin Panda, a scientist in the field of circadian rhythm. In this podcast, I will talk to experts in many different fields about science, health, and human performance. You'll also find out why circadian rhythms is my favorite topic. I hope you enjoy the podcast and thank you for listening. Welcome to another episode of Performance Around the Clock, and I'm super excited to have two scientists from the Buck Institute of Aging Research to talk about aging interventions, and everything in between. And we have Professor John Newman, who has a joint appointment between Buck and UCSF
That's
Medical
right.
School.
Yep.
And John is also an MD PhD, so that means John is one of very few people in the US who actually do both, who have been trained to do clinical research and also basic science research. And being from India, what I see, MD PhD program is the secret sauce of biomedical research in the US. It actually helps us grounded in what is relevant in clinical and translational science. And we also have Brianna Stubbs. She is the director of translational research at the Buck Institute. And again, as I said, I guess, Brianna, you are trained as a basic scientist.
I actually studied preclinical medicine, so I was planning to be an MD and then took a pivot to do my PhD because I wasn't in a formal MD PhD program, and then I didn't actually go back and finish the MD. So my background is, I thought I'd be a physician, and now doing clinical research is kind of backfilling into that passion, working with people and also working with science.
Yeah, excellent. So we really have people who are really dedicated to improving human health, unlike me, who is actually trying to improve mouse health. Okay, John, you have a long track history of research on ketone bodies. And these are the important metabolites. And can you tell us what are ketone bodies, how are they produced, and when they're produced?
Sure. So ketone bodies are how I got into studying mechanisms related to aging. I guess we'll talk about why ketone bodies are related to mechanisms of aging. And this is all part of my grand plans as a geriatrician. Like you were saying, MD, PhD geriatrician. There's not very many of us who do basic science of aging and are practicing clinicians to try to figure out how basic mechanisms of aging, which we know so much about now, help to drive the conditions and diseases that affect older adults and that affect the older adults they take care of in the hospital. And not just causing diseases like Alzheimer's disease, which is a big interest in our lab, but also these complex multi-system geriatric syndromes like delirium and frailty, which are in some ways almost too complicated to understand by kind of traditional, like one molecule, one pathway, one tissue systems, but are really systems biology problems that make a lot more sense if you think of them as being driven by mechanisms of aging, working in every system of the body to make someone frail or make someone get delirious. But the piece of that complicated puzzle I kind of latched onto when I was a postdoc with Eric Verdon, who's now also at the Buck Institute, were ketone bodies. At the time, ketone bodies were new to me too. And I knew a little bit about how metabolism and aging are so closely intertwined, and that goes back 100 years, right, to the very first experiments that demonstrated that you could manipulate aging in a laboratory, which were studies of caloric restriction with rats back in the 1930s, and showing that if you started to restrict how much they ate compared to letting a rat eat whatever it wants, that gradually they would live longer and longer and longer the more that you restricted their food, up to the point that you restricted too much and they started to starve. But since then, we've learned so much about how these metabolism pathways and nutrient signaling pathways that switch between the fasted and fed state regulate mechanisms of aging. And probably by switching us between modes when we're in kind of a state to go places and do things and grow, versus a state to do maintenance and to repair our proteins and repair our DNA. And that repair state is good for aging, and metabolism regulates it. So the aging field is really interested in these signals that mediate that switch between the fed state and the fasted state. And ketone bodies are one of those signals. So these are really, really fascinating metabolites. Your body makes them. All of our bodies make them all the time. They're one of the ways that our bodies access fat for fuel. So they're small molecules like sugar, but derived from fats rather than derived from carbohydrates. Your liver makes them mostly from fats that are mobilized from your adipose tissue. And then they circulate in the blood to whatever tissues need energy. And they're taken up by neurons in the brain, by cardiomyocytes in the heart, by skeletal muscle cells, all over the body, where they can be readily oxidized to generate acetyl-CoA and ATP to fuel cellular function. Ketone bodies have long been thought to be important to people because they're really how we survive starvation. Because most of the energy reserves in our body is in our adipose tissue. If we couldn't access that, we would only survive for a couple of days without eating. So this is the evolutionary thinking of why the ketogenic system evolved. Like, we needed to be able to survive longer than that in order to survive in the wild. So ketone bodies are how we access the energy in our adipose tissue stores so that we can fuel our bodies and survive without running low on glucose and without turning our muscles into ATP.
So then you said feeding and fasting cycle. So ketone bodies, I guess, are produced during fasting. And how many for an average human?
How long does
How many hours of fasting will
Yeah.
elevate ketone body? And before that, maybe we should wind back and ask. I might ask you how much ketone bodies are typically present in blood throughout the day when I have my 3 meals with 14 different snacks, and then after my last meal, how long it will take
Yeah.
to see that ketone body going up without any exercise. Just let's start from without any exercise, just
Yeah.
me lying in the bed and
Yeah.
dreaming about ketone.
Well, what was your last meal that you ate before you started your fast? Because that would be important.
Yeah, I suppose
too.
say I finish at 6 p.m.
Yeah. Did you eat pasta or did you eat a steak?
Yeah, so
And how full were you at the end of
Yeah.
I was really full. Like typical Western diet with 3-course dinner and 4-course dessert and ice cream and all that stuff. So, let's see.
I mean, without any activity, and assuming that you had full carbohydrate stores when you started the fast, it's going to take maybe a couple of days, maybe even as much as 4 or 5 days of fasting, to really start to elevate ketone bodies into what we would consider kind of a true state of nutritional ketosis. So to kind of provide some reference ranges, right? In some big cohort studies like the UK Biobank, they can measure ketones in people's blood, and they're very, very low, but present. They're always present at that low level of maybe, 0.01 to 0.1 millimolar, so low to trace. And that can go up to tenfold or even more, 1 millimolar up to 5 or 6 millimolar during a really prolonged fast. So 5 or 6 millimolar is very, very profound ketosis. And importantly, that's distinct again from diabetic ketoacidosis, which is what a lot of physicians have heard of. And, that's a medical emergency. If you have ketones, they tend to be more like 8, 10, 12, or even up to 20 millimolar. And that really upsets the acid-base balance of the body. And that's not optimal. That's not what we think would be a physiologic range. I think it was Krebs actually who defined a physiologic range as about 5 or 6 millimoles of ketones during fasting, based on back in the day when it was ethical to starve people for days and days and days and measure their metabolism and just give them one sodium tablet a day and a liter of water. That was back in the 60s, the kind of seminal studies of ketone metabolism, and so they saw that people would take a few days to get going, but then kind of ramp up and reach a steady state of around 5 to 6 millimoles. So that's sort of max threshold. If you were just starving for weeks at a time, your body would sit around there. But typical person, if you did a 24-hour fast and you were up and around and maybe you'd eaten a low-carb meal the night before, I used to do 36-hour fasts, and by the end of that, I could be in ketosis. So it really depends on the individual.
So what is the lower threshold that you would say that this is?
Yeah. Well, it's up for debate. It's off for a long time. It's been considered to be about 0.5 millimolar, so for adherence to a ketogenic diet. But when you start to sort of pull on the string and find out where that value came from and what evidence supports that, it's actually kind of, it was a line in an arbitrary line in the sand. And actually, John and I collaborate with Dr. Jeff Volek, who's at Ohio State University, and along with Steve Finney, they did a lot of the early work on ketogenic diets, and they had proposed that 0.5 cut point, and they're actually walking it back a little bit now, and they think that therapeutic or useful ketosis could be even 0.2 millimole. And it's interesting when you start to see ketone concentrations being measured in studies of various drugs like SGLT2 inhibitors, and they have 0.15 millimole, so only just over that barest minimum of elevation, and those are ketones are at that concentration are a possible driver of some of the downstream benefits of those drugs.
So
And there's a couple interesting things there that are actually driving how we're thinking about how to translate ketone biology into the clinic. So one is that ketone bodies are the single molecule, certainly the single metabolite in the body with the largest dynamic range. 2 orders of magnitude.
what about lactate?
Well, even lactate doesn't get as high as ketone bodies can when you're starving. So biggest dynamic range to play with. And 2 other interesting things that make figuring out how to use them therapeutically more complicated than it seems. So one is that the blood levels that you're measuring, remember that ketone bodies are burned for energy, and that's largely their fate. They're not cleared through the kidneys, mostly like drugs, or they are oxidized in cells all over your body and literally turned into carbon dioxide. So the levels in your blood represent the excess that's accumulating beyond what your body can burn at that moment. So you can start to drip in more and more ketone bodies into someone, and their blood levels won't start to rise until you've saturated how much they can burn. So we know that some cells in the body, like heart cells, are really avid at pulling ketone bodies out of the blood. So you may see physiological changes without actually changing blood levels if more ketone bodies are available. And we think that's what starts to happen even in some natural conditions like heart failure. Your body starts to make a little more of the regular 24/7 day-to-day ketone bodies. Barely enough to notice changes in levels, but enough to notice differences in how much cardiomyocytes the heart is utilizing, for example. So the third thing that's interesting is, for a long time, everyone ignored the fact that our bodies do make a little bit of ketone bodies all the time. And it's hard to measure exactly how much because of that question of, the blood levels are just the balance between what you're making and what you're using.
Almost like blood glucose, because
Yeah.
with 100 milligram per deciliter, we have only 5 grams of sugar in an average person like me.
Right.
But it's in a constant flux.
Yeah, exactly. So that flux can change, and we don't really understand yet. This is something that we're studying, and that other people too. How much that flux changes differs person to person, changes across your lifetime. There's some hints from UK Biobank, for example, that older people may start to make more ketone bodies, and is that adaptive? Is it a way they're trying to cope? Their bodies are trying to cope with some of the changes from aging. And we know that in heart failure, for example, people do make more ketone bodies as an adaptive protective measure. So a lot of really interesting things about these, very boring sounding, what are the blood levels and how much do things change, but really fascinating physiology.
Yeah, so going back to the original question, suppose say I had a standard diet.
Yep.
And then maybe for the first 6 to 10 hours, my blood ketone might remain in 0.01 or 2, like depending on which sensor you have, you may not even detect it.
Yep.
And then after many hours, then the liver will start to break down fat from the liver and make some ketone and release them, and other organs will grab them.
Yep.
And then maybe after 24 or some hours, my stored fat will begin to break down, and so that's why the fat actually goes from the fat cells to liver, and then liver makes it. So fat cells don't make significant amount of ketone.
Right.
Fat cells themselves don't make ketones. It was thought for a really long time that the liver was the only site of ketone production, but recently, obviously the kidney is a secondary site as well, but people have started to look in the heart and in the brain itself, in astrocytes, I think, and seen, and actually in the gut as well, a few sites of local ketogenesis where it's not, ketones are being synthesized, but for use in that local microenvironment. So astrocytes making them for neurons, heart, I can't remember what cell type it is in the heart, but again, some minor sites of local production. But in terms of, thinking about the big picture, the liver is the main, most important primary driver of what we see in the blood.
And then when it comes to ketone, these are 2 or 3 different types, right? Beta hydroxybutyrate, and let's go into a little bit detail.
Yeah, ketones are awfully named because they're not actually ketones in a chemical sense, but the family of ketone bodies.
Acetoacetate is a ketone, isn't it? I thought it was beta hydroxybutyrate. It was a carboxylic acid.
Okay, let's debate this.
Yeah.
But there are 3 molecules that are considered the ketone bodies: beta hydroxybutyrate, acetoacetate, and acetone. And yes, that is nail polish acetone. Acetone is a breakdown product of acetoacetate. So really there are 2 ketone bodies, beta hydroxybutyrate and acetoacetate. They're freely interconverted in the liver and in many tissues. The enzyme that goes back and forth between them does so based solely on the abundance of the 2 products and the energetic state of the cell.
It's a redox-linked reaction, which is important for, the physiologic downstream effects and the ratio between the 2.
So they're slightly different in that they have slightly different protein binding affinities when we start talking about the signaling activities of ketone bodies. And since acetoacetate is closer to acetyl-CoA than beta hydroxybutyrate, so if your neurons take up ketone bodies, they'll turn beta hydroxybutyrate into acetoacetate and then on into acetyl-CoA.
It's important to note that almost every single clinical study that looks at ketone bodies only measures beta-hydroxybutyrate because it's the most stable, it's the easiest to measure. Acetoacetate's really unstable because of this spontaneous decarboxylation to acetone. And so when you're trying to collect blood samples and protect and stop against degradation, then it's just not, you can only measure it with mass spec. There isn't a handheld device, and then it degrades very rapidly in samples. And so I think a big gap in the literature right now is that we focus on the beta-hydroxybutyrate, and we actually don't know what's going on with the acetoacetate. And even honestly, in vitro and in vivo preclinical work, people tend to treat with just BHB and not the 2 in a ratio, which is not really physiologic. But again, because when I was doing my PhD, we had some lithium acetoacetate salt, and it would just it just degrades, and it's not very easy to use for experiments. So there's that side, one big part of the equation, the BHB acetoacetate balance, we just don't really address the acetoacetate part of the picture at all.
And this is going to get important when we start talking about how to translate ketone bodies into therapeutics and use them exogenously, because the 2 molecules do have slightly different functions. Peter Crawford, for example, showed that in the liver, acetoacetate, beta hydroxybutyrate actually have opposing actions on the activity state of Kupffer cells, of the innate immune cells resident in the liver. One makes them more anti-inflammatory, pro-fibrotic, and one of them makes them more pro-inflammatory. And then there are some cell types that don't express the enzyme that interconverts them. So those cells can't use beta hydroxybutyrate for energy, but they can use acetoacetate for energy. And this is also kind of actually a cutting edge of ketone biology research. Is how are these 2 molecules different? For decades, they were thought to be they're ketone bodies. They're interconverted. They must basically be the same, but they're clearly not. And when you start to design therapies, molecules that will deliver ketone bodies exogenously, you can design molecules that will deliver more beta hydroxybutyrate or more acetoacetate or even exclusively beta hydroxybutyrate. And those may be better for different contexts.
Well, a really nice example of that is Dom D'Agostino at UCSF, University of Southern Florida. He ran an experiment where he was looking at central nervous system oxygen toxicity and the ability of ketones to prevent against seizures when exposed to that sort of extreme environment. And a purely acetoacetate-based ester was very protective against seizures, and a very purely beta-hydroxybutyrate-based intervention was not protective against seizures at all. So that really emphasize that point, that these 2 types of molecules aren't behaving in the same way.
Okay, so let's dial back. You said that two things. One is it's very difficult to measure beta hydroxybutyrate.
Acetoacetate, BHB, beta hydroxybutyrate, is quite easy to measure.
Quite easy to measure, but the commercial over-the-counter meters that are many people buy, that's detecting mostly beta hydroxybutyrate,
Yes.
BHBA. How do you measure acetoacetate reliably?
Yeah, so when we collect the blood samples, we have to, preserve them instantly, like precipitate out the acetoacetate. So you conjugate it to something, and then you use mass spec to measure that. So you have to lock it in a measurable form, and then you have that stability, and you can use mass spec to analyze it.
So if you take total ketone bodies,
Yeah, what's the ratio of
what
the two?
fraction?
Yeah.
So for example, suppose say I have fasted 4 days and my total ketone bodies is 5 millimolar.
So it's interesting because at the lower level, so when I wake up in the morning after just a regular night, they'll be in a sort of 1-to-1 ratio. At the lower concentrations, they're in an equal ratio. But as concentrations of total ketones increase, beta hydroxybutyrate is the more abundant ketone body, and it can get up to like a 4-to-1 ratio skewed towards beta hydroxybutyrate.
Okay.
But you're still missing, a quarter of the picture if you don't measure the acetoacetate.
Yeah, but that quarter can be actually much more important.
Potentially.
So let's talk about what ketone bodies do and why they have become so important for geroscience. So what are the functions on energy metabolism? Because that's how we began, that there's a fasting fuel.
Maybe I'll take energy and you take signaling.
Sure.
And then you talked about the Kupffer cell immune cells. Okay, so we can talk about the immune cells. And then signaling and maybe its impact on the chromatin or epigenome.
Yeah. So as John mentioned earlier, people always thought about ketones as a fuel to sustain starvation. And that's a main function that they have in the body. During aging, mitochondrial function breaks down. We get less sensitive to insulin. Metabolism starts to change in a way that can adversely affect end organ function. So the classic example is in the brain, where if you do FDG PET imaging and look at glucose metabolism, you can often see depressed glucose metabolism in the brain years before someone goes on to develop symptoms of dementia. And so Stephen Cunane and others up in his group have proposed that one of the drivers of Alzheimer's and dementia could be brain energy starvation because the brain is less able to metabolize glucose. So in the context of aging, having ketones as a replacement fuel that is not only entering the cell through a different pathway, it's not insulin dependent, it's coming in through ubiquitously expressed monocarboxylate transporters, and then also is entering the Krebs cycle and that like end ATP generation machine through a different pathway to glucose, bypassing potential degradation in those pathways. It's a really powerful way to kind of restore the energy to those end organs. So he ran a really nice study where he provided people with a ketogenic supplement for 6 months and then looked at the brain energy state both before and afterwards, and providing ketones as a supplement reduced the deficit of energy in the brain. It didn't completely restore it, but it went from, the brain was this energy starved, and then the ketones sort of topped it up a little bit. And they think that was why ketones were helping for brain function.
How do you measure energy starvation in a human brain?
Yeah, you'd want to talk to Steve Cunane to answer this question, I guess. My layperson's summary of it would be they're using the labeled glucose, labeled acetoacetate. Interestingly, it's acetoacetate and not beta hydroxybutyrate, but they're able to see visually using this label where in the brain is sort of lit up in terms of that fuel being metabolized a lot. And so if you took a healthy brain, you'd be able to see various areas lit up with glucose metabolism, and then you have your diseased brain and you can see that the areas are less lit up, right? So there's a visual, and they can quantify that as well. And so then you can see when you add in the ketone supplementation that the amount of light and the restoration of the metabolism in specific brain areas.
Yeah,
and this technology is getting more and more sophisticated every year. We are not specialists in this, but now there are groups even going beyond labeled uptake and PET scans to look at distribution of glucose. You can actually do in vivo, meaning in a person's brain, MR spectroscopy metabolomics. So to measure in relatively small spatial, like, square or cubic centimeter kind of areas, the levels of a bunch of different metabolites, including ATP and glucose and lactate. So you can get like real time, very detailed information on the metabolic state of the brain these days.
Oh, this is really interesting because if you go back to the history of ketone for therapeutic use, it was actually initially for
Epilepsy.
treatment-resistant epilepsy.
Right. Yeah.
So what happens there? So how does
Well, that's an interesting one, because actually people have been like struggling to tie down exactly what is the mechanism of action of ketones in epilepsy, and the things that have been proposed range from an effect of the ketogenic diet on the gut microbiome, which was a leading contender more recently, an effect of ketones on the GABA and glutamate balance in the brain. And actually, you should talk about this because John's run some really beautiful studies looking at neuronal spiking and in like a mouse seizure model as well. So yeah, I'll pass it back to you at that point. But number of different mechanisms, including cerebral vasodilation and increasing cerebral blood flow. That also happens in the heart as well. So, number of different ways. And actually, I guess that's one of the reasons why ketones are so interesting to us, because they're not just working on that single pathway. There's multiple mechanisms of action that are relevant in multiple end organs as well. But you should talk more about your seizure work, because that was very relevant.
Yeah, and this is a good segue to talking about the signaling roles of ketone bodies too, and maybe zoom way out first and just remind people that ketogenic diet is not the same thing as ketone bodies, is not the same thing as the energy stuff we're talking about. Like ketogenic diets, which have been used for epilepsy for over 100 years and arguably even longer, right? So these diets are very complex things where you're stimulating the body to make ketone bodies by reducing the carbohydrate intake of the diet.
how much?
A lot.
Yeah.
Enough to stimulate ketone bodies.
A typical cutoff is like 50 grams a day, they suggest.
Yeah. And in
50 grams of
Yes.
carbohydrate.
Yeah, like an apple, perhaps.
Okay.
and
So
the more you restrict carbohydrates, the more ketogenic the diet is.
So let's pause there, because this is very important. When we see in the popular media, there is ketogenic everything, including in a conference I found ketogenic wine.
Oh, yeah. Well, that'd be like a low carb wine, right? I don't think they put any fat in that wine. Yeah, dry wine.
But the point is, when you say ketogenic diet, it's the diet that actually primes the body so that it produces its own ketone.
Yeah, it's a pattern of eating, which mostly involves lowering carbohydrate intake. And then if you ask dietitians like Jeff Volek, who do this clinically or do this for research studies, they like to talk about a well formulated ketogenic diet, which means that if they're doing that in the healthiest possible way, it's going to involve eating a lot of fat, but healthy fats, and it can even be plant-based. And you don't want to overdo the protein intake, normal protein intake. So you reduce the carbohydrates way down, but try to keep the other elements as healthy as possible.
So the bottom line is, if somebody really wants to do ketogenic diet, should actually talk to a dietitian. And
Absolutely.
Yeah.
It's gotten increasingly hard to do the ketogenic diet well, because there's so many products out there claiming to be compliant with that diet. And it's also because it's a really powerful metabolic intervention, evidenced by the fact, as you said, that it was used as a medical treatment for seizures. And, it's now being looked at as a medical treatment for various other conditions, including diabetes, right? There are some conditions where ketogenic diets are very beneficial, but average Joe on the street, you would want to be doing this in conversation with your doctor. A very important risk factor is changes in blood lipids that can happen on the ketogenic diet, and you'd really want to have pre-diet blood work so that you can check in a few months' time whether things are starting to look wonky, because they're just it's powerful, and it's not just a thing that you change and has no other consequences, and it can be hard to formulate it right. And getting advice on what type of fats to include, from fish, from nuts, from avocados, not just putting a whole stick of butter in your coffee, which is what you see some people do, right? There are so many ways to do the ketogenic diet badly and so many products and gurus out there on the internet that it can kind of be hard to sift through the noise. So I'd recommend people not only talk to a doctor, but do talk to a dietitian so that you can construct a ketogenic diet in a way that's healthy, and then monitor yourself for any adverse events.
Because in mouse literature also, it's very difficult to do a ketogenic diet, even for a little mouse, because when you look at some of the popular ketogenic diet, they go up to maybe 60% fat and then 15 to 20% protein.
Yeah.
And if you give a mouse 60% fat, 15% protein,
It's
and then
not
the rest
ketogenic.
carbohydrate, that's actually pathological. That's the
The
diet-induced
high-fat
obesity.
diet, yeah.
Wonderful.
Exactly.
Well, that's important to say, right? That if you see a high-fat diet in a rodent paper, that might not necessarily be a ketogenic diet. In a human, a ketogenic diet often is high-fat, but in a mouse, high-fat does not equal ketogenic diet.
Yeah, so that's a very important distinction, because up to 63% fat, the mouse is actually because it's pathological, so the mouse becomes obese and
Yeah.
has heart disease and all
Yeah.
that. And then only when you go up to maybe 70-80% fat, but reduce the
higher.
carbohydrate,
Yeah.
that's much more important, because reducing carbohydrate in mouse chow to
Yeah.
less than 10%, I guess.
Yeah.
Um,
So
but
for all the mouse biologists out there, if you're interested in doing ketogenic diet studies with mice, take your time and learn the literature, because there's a lot of ways to do it that you will regret after you
You'll get
finish your 3-year experiment.
Some angry reviewers' comments, maybe.
And my postdoc was learning all the different ways that I was about to do a ketogenic diet study the wrong way, and eventually I think I figured out the right way to do it. But you need to really restrict carbohydrates in mice, because their metabolic rate is so fast.
Yeah.
So, but you can actually feed a mouse a diet with 0 carbohydrates, and they're fine. They're fine for years.
It's very difficult to do that study, because most of these diets are almost liquid, and you got to change the diet every single day.
So the consistency and the fat content and the palatability become really important. Keeping that balance of protein intake, because if it's too high, you can make it less ketogenic. And also, really high protein diets are bad for longevity in mice. But if you make it too low, now you're not actually studying ketogenic diet. Now you're studying protein restriction, and there's a whole literature about how protein restriction, methionine restriction affect aging. So you really have to very carefully design a well-controlled ketogenic diet for mice. And in our lifespan study, we used a diet that was 90% of calories from fat, about 10% from protein, which is a normal amount of protein for an adult mouse. Most of us, slight aside, but a pet thing for me, most of us mouse biologists use diets for older mice, like middle-aged mice, old mice, that are really meant for young mice, for growing mice, which have probably too much protein to be normal for an adult mouse. So we use 10% of protein, which is about right for an adult mouse. And then the other thing is that in humans, people use ketogenic diets for weight loss, but that's not generally the case in mice. Most mice really like the soft texture, they really like the high fat, and they will overeat a ketogenic diet and get very obese. So the key technical challenge in longer term studies of ketogenic diets, or if you really just want to focus on the biology of the ketogenic part of that, is avoiding the obesity.
I joke that John's the father of the cyclical ketogenic diet that some of the gym bros do, because his paper had them on and off and on and off. And I don't know whether I've seen anyone else do that. They might have done since, but no one had done it before.
I remember that paper in Cell Metabolism.
Gym bros around the world rejoiced.
No, I guess this is very important, that this also raises another point, that many of these studies in mice, usually, you don't want to try any new dietary study in human and start to look at effect in different organs. Usually we start in mice.
Yeah.
And it's also important to read between lines. What are the composition of the diet? And as I'm reading more and more papers, I'm also figuring out that some of the macronutrient proportion may not be right for what we are studying. So this is very important for people who like to deep dive into mouse literature or human literature. Okay, so we're talking about the seizure and epilepsy.
Yeah, and that ketogenic diets are this really physiological, complicated thing with different nutrient ratios and all of that. And one component of that is that it stimulates production of ketone bodies. So ketone bodies are not the same thing as a ketogenic diet. And as we start to talk about exogenous ketones and ketone esters, ways of administering ketone bodies, either experimentally in mice or in the research setting in people, or in other settings with people. It's not the same thing as a ketogenic diet in a pill. It's one aspect of a ketogenic diet, but it happens to be a very biologically interesting aspect.
do like to put out the big disclaimer that if you're looking for weight loss, that exogenous ketones are definitely not ketogenic diet in a pill, because they're often badly marketed as, a shortcut to weight loss. Exogenous ketones are often badly marketed as a shortcut to weight loss, which they are not.
But I guess I can say that once I bought, somebody gave me some really bad
Bad-tasting.
ketone,
Mm-hmm.
I don't know what, but anyways, so the point is, once you drink that, you don't feel like eating for the next 4 or 5 hours because it leaves such a bad taste.
Well, in my PhD, I studied some of that, and I looked at ghrelin, the appetite hormone, and it's actually suppressed compared with the same amount of calories as carbs when you take ketone drinks. So there might be something there other than just the bad taste.
Yeah. But anyways, the bad taste was
But it's pretty bad, yeah.
But if you're thinking about enrolling in our clinical studies, I want to reassure people that the ketone ester that we were using in our studies with older adults has a much more neutral taste.
Yeah. No, I totally agree, because over the last 7-8 years, I have seen how the ketone drinks have evolved. Initially, they tasted like
Nail polish remover.
paint or nail polish, and now they're
Yeah.
more palatable. So going back to okay, so these are drivers of energy, and we don't know exactly how it treats epilepsy, but that's for debate. And as long as it is treating, that's good enough.
It's the epilepsy, what we know about the mechanisms of ketogenic diets in epilepsy is maybe a good analog for aging too, because it's not that we don't know the mechanism, it's that epilepsy is a heterogeneous set of disorders driven by a bunch of different underlying mechanisms, and different parts of the ketogenic diet probably are effective for different individual subtypes of epilepsy. When you start to dive into the epilepsy literature in mostly mouse models, and where you can be really specific about testing individual mechanisms, for some models of epilepsy, it's the carbohydrate restriction. For some models of epilepsy, it's the microbiome effects. For some models of epilepsy, it is the ATP generation from acetoacetate, but it's different for different parts. And that's like aging. So that pleiotropic nature of the effects of ketogenic diet and of ketone bodies makes them interesting as potential gerotherapeutics targeting different aspects of aging, because different parts of ketone body biology hit a bunch of the different mechanisms that are involved in aging and age-related diseases. And so if you have a complex disease that has a bunch of loss of ability to burn glucose for energy, plus abnormal chronic inflammation, plus epigenetic changes, it's tempting to think that an agent that impacts all three of those things might wind up being effective. And that's part of the thinking behind testing ketone bodies as in a geroscience context.
Yeah, so this is interesting, because we have been always looking for a intervention, whether it's a lifestyle, drug, or supplement, that should not target only one aspect of aging, but multiple aspects, so that we can fix a lot of things. It's like taking your car to a car shop, and if the car shop is specializing in all aspects of your car, that's much better. You leave the car for a day and then you come back, most of the things are fixed. Instead of taking only for oil change in one day and then going to another tire shop for another day and brakes another day.
Yeah.
So coming back here, one thing we discussed, you pointed out, is the effect of ketone bodies on inflammation. And can you expand on that? How immune cells use ketone bodies, or how ketone bodies affect immune cell function?
Yeah, and there's a few different mechanisms that people have worked out in mice and in cells. This is also a great topic to talk about how important translational research is, because of most of what I'm about to say, we know really well in mice, but we actually don't really know at all of how these mechanisms work in people, and that's what we're starting to try to find out. But there's a really interesting literature that started out of Deep Dixit's lab at Yale about how beta hydroxybutyrate, in particular, inhibits the activation of the NLRP3 inflammasome. So inflammasomes are these kind of master regulator processor molecules in our innate immune cells. And in response to different forms of inflammatory stimuli, they're these complex multi-component molecules that will assemble into a big machine and then kick off an inflammatory response by cleaving and activating and releasing certain cytokines. So they're sort of like central processing units in immune cells, like deciding, based on everything I'm seeing, should I become inflamed? and that's a good thing in the proper context, but it can become a bad thing if you get chronic maladaptive inflammatory activation in aging. So it's really interesting that beta hydroxybutyrate inhibits the assembly and activation of these inflammasomes, especially in peripheral macrophages. And Deep Dixit's group went on to show that this is important in at least some inflammation-related disease models in mice, like gout. But it's so interesting because the NLRP3 inflammasome and that innate immune inflammatory activation seems to play a role in all sorts of diseases of aging, in dementia, in type 2 diabetes, in cardiovascular disease. So it links beta hydroxybutyrate through inflammation to a whole bunch of diseases of aging.
So you said macrophages. So these are the ones that go and gobble up sick cells.
Yeah.
And you also used another term when we were talking about the liver, the Kupffer cells.
Mm-hmm.
So the Kupffer cells are the
Yep.
local macrophages.
Right. Yeah.
So there are also tissue resident macrophages in kidney and brain.
Mm-hmm.
So the idea is maybe this beta hydroxybutyrate might be putting a kind of I won't say break, but a much better control system on the inflammation, so that after it is,
Mm-hmm.
you got to push the gas, but at the same time you should not push it too much. You should also have a break. So it's almost like the break on the gas, break and gas.
That's the idea. But when it comes to, and that's what seems to be the case in mice. Now these tissue resident populations, like microglia in the brain, or Kupffer cells, they all do respond a little differently to various stimuli. So actually, it's an active question that we're studying in our lab, whether microglia in the mouse brain or human microglia respond similarly to ketone bodies in terms of inflammatory activation. And you can look for some work to come out very soon on that from one of the postdocs in my lab. But there's other ways too that ketone bodies regulate immune activation. And again, this has all been worked out mostly in mice, but not so much in people. Many immune cells, especially T cells and T regulatory cells, are sensitive to the metabolic state. So if you change their metabolic state, you change their activation, you change their phenotype. And ketone bodies, by converting them kind of away from a glucose-dependent metabolism and towards a fat ketone-dependent metabolism, changes how they respond to inflammatory signals. So that metabolic state, that metabolic regulation Brianna was talking about, it's not just about general energy production, but it actually changes the way that some cells function in the body too. But a big frontier is, what about in people?
Yeah. Yeah, and there's been John Little's group up in Canada has been the first people trying to look at that, and they've been using blood cells that they collect from people who drink ketone drinks and doing in vitro experiments using fresh blood cells, and they haven't necessarily all been coming out looking the way that we expected based on the preclinical data. So I'd say it's very much still an emerging field, and as John mentioned, we're looking at it in our upcoming studies.
Yeah, I understand. Like after dabbling in human studies, I realized that humans are so different one from another, and then it's really difficult to figure out what they did in the last 5 days or even last night to laugh at, because if they had sleep disruption, for example, then the immune system is very different.
Right.
Yep. At least with things like sleep, the wearable technology is getting an awful lot better. I think it'll be interesting to see in terms of measuring metabolism, right? We have continuous glucose monitors. What's going to develop in the next 5 or 10 years is going to give us more continuous insight into people's metabolism, and then pairing that with wearable devices. And, if we can properly tag data, this was a true, you woke up and you went to the bathroom, or you woke up and you were moving around and you were stressed, and then what happened in metabolism, or maybe before you got up in the middle of the night, you had a low blood sugar moment, and there's some linking going on there. So if we can start to link more continuous data streams together, that's going to give us better windows into health. And again, we have some collaborations and some projects that are starting to try and link different data streams like this together to help us to understand more deeply health trajectories.
Yeah. So the bottom line is what you said. in addition to macrophages, T cells.
Mm-hmm.
We are not talking about B cells. T cells, depending on whether they are using glucose as energy source or ketone as energy source,
Mm-hmm.
they can respond to infection or immune activation differently.
Yeah.
So that also means that if I'm fasting for a long time
Mm-hmm.
and I had a infection or I caught flu, my body might react very differently.
Well,
I'm not saying that it will be better or worse.
well, actually, the experiments from John Little showed that in people who were obese and so had higher basal chronic inflammation compared with in young healthy adults, ketones actually boosted the immune response to an LPS stimuli in vitro. So it was completely the other way around to what we expected. So, but then in the young healthy, it was different in depending on the basal state of the donor of the
So
cell.
that's why I'm saying that
Yeah.
we don't know whether this response will be good or bad, but it will be different
Yeah.
from
Yeah.
what you would expect with glucose as an energy source.
Yeah.
And so we talked about inflammation and energy source and energy usage. There is also effect of ketones on epigenetic regulation.
Yeah. Yeah. And this is a real cutting edge area that we have a few hints of why it's important, but it's fascinating that it happens at all. And the extent to which it happens is pretty remarkable. So ketone bodies clearly regulate the epigenetic landscape in cells. And they do that through a few different mechanisms, all of which kind of interact with each other to change the epigenetic landscape.
So when we are talking epigenetic landscape, is it modification of DNA or the chromatin or the histones?
Yeah, great
What
point.
we are talking about?
So I'm going to mostly talk about histones, so histone tail modifications, but things that happen on histone tails happen on other proteins also. But we know a little more about the histone tails for ketone bodies than we do for other non-histone proteins. But then of course there's secondary effects, right? So if you change the histone tail modifications, you might be bringing in other enzymes that change DNA modifications. But we'll focus on the histone tails. So first. Beta-hydroxybutyrate inhibits the activity of one of the major classes of histone deacetylases. In other words, making histone deacetylases work less efficiently and less able to deacetylate histone tails. So overall, promoting increased acetylation of histone tails. Beta-hydroxybutyrate ketone bodies in general also provide the substrate for acetylation, right? Because they're oxidized to acetyl-CoA. And so it increases the pool of acetyl-CoA, and that's another way that it can promote protein acetylation. But then a few years back, some other scientists discovered that beta-hydroxybutyrate itself is actually covalently attached to proteins, including histone tails, as a post-translational modification. So that's called beta-hydroxybutyrylation.
There's a mouthful.
Yeah, it's a mouthful, and so up there alongside phosphorylation and acetylation and
Methylation.
And methylation. Now you have beta-hydroxybutyrylation. That happens on all across the genome, and there's a nice paper linking it to sites of recent gene transactivation. So sites that were recently opened so that transcription could be upregulated, and those histones get more beta-hydroxybutyrylated. When the stimulus to change transcription is fasting, right? So fasting, you turn on this whole new transcriptional program, and those genes, their histones, get beta-hydroxybutyrylated. But it's probably the case that other proteins get beta-hydroxybutyrylated too, but that landscape hasn't been fully fleshed out.
And because histones are so much histones in the cell, so it's easier to detect that. Whereas other proteins, compared to histones, there may be 100,000-fold less. So it's the sensitivity becomes an issue.
Yeah. And we don't know what it does. So we don't know why histone tails get beta-hydroxybutyrylated. We don't know what other enzymes involved in chromatin regulation that interacts with. We don't actually know, does it change? Does it have a direct effect on changing transcription, or is it just there? We really don't know what the biology of beta-hydroxybutyrylation actually is. And that's just the very tip of the iceberg in this field of metabolic regulation of epigenetics, because it's not just beta hydroxybutyrate. Anyone out there who remembers like undergraduate biochemistry and the Krebs cycle and basically I've learned that every metabolite in the cell of which there is a CoA form, succinyl-CoA, malonyl-CoA, acetoacetate CoA, that metabolite.
I think there are 2% of proteins who have CoA, or 2% of enzymes who have CoA, or something like that. I had read in some textbook long time.
Yeah, it's a lot of, it's a common way of facilitating reactions between molecules, but you wind up with a lot of metabolic molecules in the cell floating around with these activated CoAs on them, and every single one of them can become a post-translational modification. So you have succinylation, you have malonylation, you have lactylation, lactate. So there's this whole smorgasbord, this whole like zoo of small metabolite post-translational modifications getting attached to histone tails. And we mostly don't know what they all do. But just imagine, like, when you're eating that big meal and then fasting afterwards, just how all these things are changing in your cell and totally remodeling the landscape of epigenetic modifications in ways that we don't understand the significance of.
So acetyl-CoA, which is the ketone body. So you take the acetyl part, you can do histone acetylation, and that changes interaction of DNA with histone so that those genes can be turned on, mostly histone acetylation. And then the CoA part can also go to different enzymes and proteins and attach to them. Acetyl part can also go to other proteins, but CoA can also go to other proteins and can either activate or turn on or turn off.
Yeah, in this case, thinking about metabolic post-translational modifications, the CoA is basically the battery that provides the energy to attach the metabolite to the histone tail. So it's an activated bond that provides that binding energy. So you can take beta hydroxybutyrate CoA and attach the beta hydroxybutyrate directly to a protein covalently.
Is it a high energy bond like the phosphate bond in ATP?
Essentially, yeah, similar idea.
Okay, so this may be another mechanism by which ketone bodies affect signaling.
Yeah.
Is that?
Well, I mean, you didn't even mention, right, that the effect via histone deacetylase inhibition was what you showed with Eric Verdin, was to affect oxidative stress via changing levels of SOD and other catalase and other elements of the detoxification system inside the cell. So there's downstream, you have the highest level epigenetic changes that are triggered by changes in acetylation or BHB elation itself unknown, and then the downstream effects, and the oxidative stress was one of the first ones that was described, but there are potentially many others as well.
And a really nice study from Omer Yilmaz at MIT who looked at production of ketone bodies in intestinal stem cells, and so that's an example of local production outside the liver. But the bottom line was that making ketone bodies in intestinal stem cells helped to maintain the stemness of those stem cells through changing histone deacetylase activity. So very nicely tying together, like, here's a signaling function of BHB that's actually necessary for helping to regulate an important hallmark of aging, maintenance of stem cell function. So these are all reasons why we are so excited about trying to study ketone biology in people in the aging context. And what can we learn about which of all these interesting mechanisms that interact with hallmarks of aging, which of them are really happening in people that we can invoke with exogenous ketones.
So I guess I'd say at this point that we had all of this biology, but no studies that had yet looked at ketone bodies as a supplemental strategy in older adults. So most of my career I'd been working on drinkable ketones, and John and Eric again had been developing different types of compounds. You mentioned like things that taste bad. These again, they're more neutral tasting, but there's the idea of an exogenous ketone is that you can consume it typically they're beverages, but you consume it and the molecule either contains ketone bodies themselves or it contains precursors that are very easy for the body to make into ketones. So examples might be the ketogenic alcohol 1,3-butanediol, which is in a lot of these molecules, medium chain fatty acids that are part of these ketone esters as well, or beta-hydroxybutyrate or acetoacetate itself. We all eat esters every day anyway, so the esterase enzymes that break apart these ketone esters are ubiquitously expressed, so we consume them, they get broken down in the gut and absorbed. Because the liver doesn't really use ketone bodies, they just pass straight through the liver into the systemic circulation. And then you've got this increase in blood ketone bodies that can typically range anywhere between 1 to 4 or 5 millimole, depending on how much of it you take and which compound that you take. But that increase can happen within 30 minutes to an hour. So you kind of bypass the need to fast for days and days and days. And from a scientific perspective, we can isolate the effects of just increasing ketones from the effects of either caloric restriction or carbohydrate restriction. So it's an interesting scientific tool as well as making it kind of translatable. But so range of compounds out there. The main goal of these compounds is to increase circulating ketone concentrations. And one of the very first experiments that was done using these compounds was as an energy drink for athletes and looking at athletic performance. And so the first few years that drinkable ketones were available, people were really just focused on this energy role and looking at human physical performance. And so there was real lack of studies in older adults and really a lack of long-term studies as well. So we couldn't find any study that was longer than 4 weeks. And obviously, if we want to apply drinkable ketones as a long-term strategy for healthy aging, then you need to extend the safety data and also look specifically in older adults who might have differences in kidney function, liver function, metabolic breakdown of all of these ketones. And so in the last couple of years, together, we've run this study at the Buck to kind of provide some of that foundational information around the safety and tolerance of drinkable ketones in older adults. And it's also given us an opportunity to start to pilot some older adult specific functional outcomes and biomarkers that we kind of, based on the preclinical data, think might change in older adults taking ketones. So we're among the first doing this work.
Yeah, no, that's exciting. So that brings back one question in my mind. So suppose say you take 2 individuals identical in BMI and only BMI, let's not, and maybe sex and BMI. One is 25-year-old, one is 75-year-old. And you put them on exactly the same diet.
Yep.
And you make them fast for X number of hours. Does aging reduce or increase the ability to produce ketone so in these 2 individuals, what will it be the same amount, the kinetics of rise in blood ketone will be the same or different?
I mean, I can speak to how people respond to drinks, which gives us a bit of an answer to your question.
No, actually, this is more important about because when we're talking about older adults, the first thing that comes to my mind is, are older adults less efficient in producing ketone? And maybe that's why the brain was not getting enough ketone. And has anybody looked at that?
Has anyone done it in people and not mice?
I think the answer is probably.
Yeah.
But I don't think it's been carefully looked at in people.
No.
Like for your question about fasting, but data from mice can and some of what we know about people can, I think, paint a picture. Probably as we age, we probably start to make more ketone bodies at a baseline level, but have a harder time ramping that up in response to a fast, for example. In mice, we actually see...
This is important. So at the baseline, the older adults may have slightly more. We're not talking about like going from 0.01, 0.02
to 1. Yeah, we're talking about going from 0.15 to 0.25, perhaps.
So let's make that very clear.
Yeah.
Very small difference.
Well, I mean, it's a big in terms of like a fold increase, but in terms of the dynamic range of ketones, as you said, it doesn't represent a big change within that dynamic range. Yeah.
So then the point is at the clinically significant level. So that's 1 to 2 or whatever.
Yeah.
Whether it's even 0.5 millimolar. So that's where older adults or even regular athletes, for example, can actually take a ketone drink, and depending on how much is okay. So let's go back and ask very simple question.
Yep.
How much ketone drink is safe?
Okay, so the body can make up to 150 grams of ketones a day during fasting. That's been used as like an upper limit guide for people thinking about designing these strategies. Studies so far have used up to 75 grams a day for about a week, and in younger adults and not found any safety issues. And our studies used 25 grams a day. So we're talking, I think that's another important point to emphasize. Compared with a drug where you might be taking a few milligrams or a few hundred milligrams of the substance a day, ketones are basically much more like a macronutrient, much more like, if I'm running a race and I take a gel, it's got 30 grams of carbohydrate, and the dosing for ketones is much more in the realm of macronutrient dosing compared with drugs. So at the doses that have been studied so far, and I imagine up to that 150 gram range, you're sort of within an amount that the body itself could produce naturally. But you can never. The one thing that's different about taking ketone drinks is you're doing it in the presence of replete other macronutrients, because normally the body would only see ketones when glucose is low. And acutely we see that if you drink ketones and increase your blood ketone concentrations, there's actually a very interesting corresponding decrease in blood glucose and also a decrease in blood-free fatty acids as well. So it's almost like in a healthy person in the short term, the body can kind of hold the energy content of the blood fixed in that short term. But people who worry about long-term use of these interventions raise the valid point that it's not physiologic to have a donut in one hand and a ketone drink in the other hand and really be like loading those systems.
It's not a balanced diet.
It's not a balanced diet. It might feel good and balanced in each hand, but it's not. So, and it's not physiologic. So, there hasn't been any safety concerns seen at the blood concentrations that are reached and at the doses that have been studied. But I don't think either of us would advocate to like stop safety monitoring at that point, because, it's still like an emerging field.
So let's go back to your point about safety monitoring and human studies. And Buck was one of the first to test these ketone supplements in humans.
We did the first study of a ketone ester in older adults specifically.
But on the top of a big body of literature using human subjects that's been going in 15 years at this point.
Yeah. So on older adults.
Yeah.
And this is really important in thinking about like geroscience interventions generally, a lot of which people are thinking about either molecules that are sold as dietary supplements or repurposed drugs that are in wide clinical use. But so the geriatrician in me knows that there's a lot of ketone drinks out there that are very safe, and they're generally registered as food ingredients or nutritional supplements, generally recognized as safe designations. Like very safe. But the geriatrician knows that anything that you do with an older adult, you're never quite sure how the older adult's going to react. And so you just want to be a little more careful. And even if you're thinking about, trying a drug that's in wide use, but using it for aging outcomes in older adults, you probably still want to do a study specifically in that population, older adults. So that's why we started very cautiously with actually looking at, so if we give this particular ketone ester to our older adults, we're pretty sure it's going to be really safe. But do we see any signals of problems in their labs, in how they react to it, in the side effects that they get? Are they getting nauseous? Are they getting headaches? Are they getting lightheaded? Blood pressure?
We also look specifically at older adult specific pharmacokinetic responses as well.
When you say older adults, it's above 65?
Yeah. And the average age of our study population was 76, so older.
And our oldest person?
Was 89.
Yeah.
So I think this is common among all medicines. So most of the medications are typically tested on adults between the age of, say, 18, 21 until 55, 60, 65.
Because things get so much more complicated the older you get.
And it's a pet peeve. Even medicines that wind up getting approved for diseases that mostly affect older adults, where the average age of the person taking it is going to be like 70 to 80, but still generally the trials that lead to their approvals are much younger and have very few older people in them.
Because recently I was looking at GLP-1 receptor agonist, and the upper limit was 65 for the age. And there are so many older adults who want to take it. So this is very important that
Mhm.
almost any drug, any supplement needs to be tested in different age group, particularly older adults.
Yep.
Yeah.
And okay, so what was the study and what have you found?
Yeah. So I got to say, Satchin, that this is why being an MD PhD is so fun and rewarding, because I started off studying ketone biology in mice and the lab and very basic science, and separately, I finished training as being a geriatrician and taking care of people with frailty, for example, in the hospital and trying to maintain their mobility and exercise. Uh, but then, I was able to bring those two things together and meet in the middle, because the things we've talked about with ketone biology, the energy supplementation, offsetting declines in glucose metabolism, the effects on chronic inflammation, these are part of what we think are the pathophysiology of frailty. And frailty affects a bunch of different tissues throughout the body. So you really, if you're going to try to intervene on it biologically, you want to use something that's going to affect a bunch of different tissues, everything from the brain to the muscle to the heart and everything in between. So I was able to, so it seemed to make sense to bring my basic science and clinical interest together and try to both see if the biology of ketone bodies translates to people, but do it in older adults with an eye towards, will it affect the biology of frailty, and will this lead to something that will lead to better treatments for frailty, which is really fun.
Yeah.
It's taking science and it's starting to apply it in a way that might make a difference in the world to a lot of people, and really bringing my clinical and basic science interests together. So,
especially because ketone drinks are commercially available, and so it's not like if we were to find something that there'd be a 10-year drug approvals process for people to maybe start being able to implement things in their own lives.
But right now, when you see your patients with frailty, what is the current state of the art standard of care?
activity, exercise, in whatever context you're seeing the person, more activity. So in the hospital, that means getting out of bed more and walking in the hallways more if they can, and removing barriers to that. In the outpatient setting, it's some sort of structured exercise program under the supervision usually of a physical therapist, and then a comprehensive geriatric assessment to try to otherwise optimize them, in part so that exercise intervention is more effective. So optimize their medications, if there's something that maybe they shouldn't be on, it's causing side effects that are getting in the way, removing that, or making sure their chronic diseases are treated well. But there's no medicines. There's no pharmacological agents for frailty.
So that's why this will be super exciting, because if they can just take a 25 grams, 50 grams drink or whatever.
Yeah.
Ideally, in addition to any lifestyle.
Healthy eating, and then that can give them that energy boost, at least to get out of the bed.
Yeah.
Because
Yeah.
a lot of adults, they actually feel very low energy, fatigued, and they don't have the.
It's interesting that you use the word fatigue, because the study that we've conducted, as John mentioned, was foundational for the study that is about to start, which will, the first study was in healthy older adults and was quite small. This next study is going to be 5 to 6 times as large and be in pre-frail adults. And we're using a composite outcome that not only looks at physical function, so we're looking at leg strength, walking endurance, and then digit symbols, which is kind of, synthesizes some cognitive processing speed and movement speed as well. But we're also using a self-reported measure of fatigue called the Pittsburgh Fatigability Scale. And we think that really bringing all of those different elements together gives us the best, way of capturing how people fall on a spectrum from vigorous to frail. And this composite score was developed by the SOMA Study of Muscle Mass and Aging study investigators. And so we've been very excited to start implementing that, because it's so holistic and whole person way of measuring function, rather than just a binary like, are you frail or not?
Yeah. So what is this pre-frail? Can you define what is pre-frail? And you said 5 to 6 times larger.
So the first study was 30. This follow-on study will be 180, and there's a 3-site study. So we're partnering with Jeff Volek at Ohio State and Jenna Bartley and George Kushel at UConn to run this study. So they'll be the recruitment will be spread across 3 sites.
So our first study was called BIKE. It was a pilot study of a ketone ester in older adults. BIKE stands for Buck Institute Ketone Ester Study. I'm mildly proud of that.
People did get confused about if they'd have to ride on a bike or not, but there was no bike riding in the bike study.
Actually, I was thinking of it.
Yeah.
Novardo is a nice place for biking.
Yeah.
It is. It's gorgeous, yeah.
Lots of hills.
So bike was actually the first clinical trial ever done at the Buck Institute. So it was a vehicle for us to build the infrastructure to do clinical trials, and also to gather some important initial information with older adults and this ketone ester, so that it's tolerable, that we didn't detect any red flags of safety concerns, that it raises their ketone body levels.
Interestingly, by a little more in the older adults than what we've seen in the younger adults. It looked like older adults had a greater response to the same body weight adjusted dose, which is something that we're planning to publish and also following up with other work as well.
And we did a lot of biology testing too, to see if we could identify, it's a small study, but still see if we could identify signals of some of the biology at work. And so things like proteomics and on plasma, and looking at the phenotypes of white blood cells, and looking at the methylation state of white blood cells, and a bunch of other things. We're still working on analyzing all of these data. The rest of BIKE is either published or about to be. But there are some interesting signals that we could see, even in this small study of BIKE, that something is going on biologically that seems to make sense from what we know about ketone bodies. So we think they're doing something.
So in bike study, there were 30 adults who
are healthy.
Was
76.
76 plus, and how much ketone they had to consume and for how many days?
25 grams a day for 12 weeks.
12 weeks. Okay.
Yeah.
So that's pretty.
Well, I mean, we went from 4 weeks, remember that being the longest study before, so 3 times as long. And then, and the next study will be 180 pre-frail, and John can describe that in a second. We designate that based on gait speed over 4 meters. We'll also be using that composite outcome as our primary outcome. We'll be extending the study to 20 weeks, so even longer than our 12-week study. And we're also planning to increase the dose so people can go up to 50 grams a day depending on tolerance. So we want to keep some flexibility with, a tried and true geriatrician method of, start low, go slow, and build people up.
But at the same time, when you're giving them, you're also asking them not to eat that donut for.
We really did soul search about if we would make diet or exercise recommendations, because we do think that combination interventions have the most potential to make a difference to people's function. But with the budget of the study, it was an NIH funded study, and the budget did not allow us to add in something that would give us sufficient control. If you're going to make diet recommendations, you have to be good at following diet and providing diet advice, dietitians, and just a lot more staffing. And the same really with exercise interventions. You either need to be tracking people's activity very, in a very valid way, and potentially having exercise physiology on staff to oversee that. And so we didn't have the capacity to do that, but we did soul search for a long time. And I think that if we had infinite money, that would be how we'd have run the study, with some additional lifestyle intervention alongside the ketone ester.
So I like that we're part of the philosophy of takeoff. And I'll tell you what takeoff is in a sec. Is a mechanistic biology study, but with pragmatic elements, where we're trying to meet people where they are, with the idea that if a ketone ester does seem like it might be useful for older adults, most people aren't going to be taking it in the context of an intensive big change to their diet and exercise habits. It'll be something they add on to what they're doing. So we decided that we want to see how the biology changes, but we really want to see the biology change in the context of people doing what they normally do. And that's how it would be most useful.
Yeah, more pragmatic.
Yeah, so it's a really interesting hybrid of a very mechanistic focused but also pragmatic kind of clinical trial called Takeoff. I'm really proud of this one.
Okay.
Targeting aging with a ketone ester for function in frailty.
Okay, that's much better than bike.
Yeah.
Bike, and then you take off.
Yeah.
Exactly.
Biking towards the takeoff. Yeah.
Biking towards takeoff. So when is this study going to begin, and how long is this study, when it's expected to end, so that we can anticipate results?
Yeah, it should be starting very soon. So we've been working like mad to get it launched over the past year, getting all the different pieces in place and across the 3 sites. We're just about ready to start screening and enrolling, and we'll try to make sure that everyone knows when we are. but takeoff should indeed.
Possibly by the time you release this, we'll be screening and enrolling. If it's not released tomorrow, then.
And then it's about it's a 5-year program altogether, and we'll probably be enrolling and having people on the protocol for about 3 years is our goal. And most of the analyses will happen after that. So start to look for results about 4 years from now.
So they will do this for 5 months, 20 weeks.
Mm-hmm.
They will take a ketone ester drink or
Yep.
once a day, and then you will monitor them for 2.5 years, or?
Oh no.
No.
Yeah. So
So the
yeah.
I
So
know the recruitment is very difficult.
yeah. So the overall study design is a parallel group, randomized parallel group, and double-blinded study.
Yeah.
So when people will be screened and enrolled, like all of these centers, Ohio and UConn are, big university hospital systems, but they're not attached to clinics specifically. They're, UConn is a geroscience-focused research unit, and Dr. Volek's lab is focused on dietary interventions. And so it's not like we have a stream of patients coming through where we can just pull people in. The other thing is that, having our gate speed criteria means that we anticipate we're actually going to screen fail an awful lot of people who are maybe too slow or a bit too fast. So
What is the criteria?
0.6 to 1 or 1.2?
Yep, 0.6 to 1 meters per second,
Yeah.
which is, you're asking what does pre-frail mean? Yeah. So the idea with takeoff is that we don't want super fit, perfectly healthy people. We want older adults who are expressing the physiology of aging, because we want them to be a little impaired in their glucose metabolism. We want them to have a little bit of chronic inflammation. We want them to be expressing the biology that we're trying to target. But we want it to be at an early enough stage to give us the best chance that a ketone ester will be able to change that in ways that we can detect in their clinical outcomes too. So we really want to try to enroll people in a window of vulnerability, and that's what the gate speed criteria is meant to capture. And it's this idea of pre-frailty, that they're slowing down in ways that you can measure. They're expressing some of the biology of frailty, but they're not too frail yet. Now it's an open question whether you can reverse frailty once it sets in. Some people think you can, some people think it's hard. But our goal for takeoff is to be a little bit more preventative, try to capture people and intervene on them when there's pretty likely going to be a good chance to change their biology and change how their body functions.
So people will be screened continuously throughout the 3 years and then brought onto the protocol. We do a bunch of testing at baseline where we do a safety check at 4 weeks just to make sure that anyone isn't going in a wonky direction with lipids and whatnot. We reassess all of our outcomes again at 12 and 20 weeks. And then after that, their involvement with the study ends. We often keep permission to contact people for follow-up, but that's not built into the study at this point.
Let's go back to the gate speed, because many listeners, they would think, okay, so am I frail, or am I okay? So can you describe a little bit more?
Yeah. So
maybe I'll qualify. No.
Maybe we could demonstrate.
Yeah.
I won't put you up to that. But so like frailty, aging is very complicated.
Yeah.
Frailty is a very complicated concept.
Yeah.
But it turns out that you can measure it relatively simply, because simple actions that we do every day, it turns out, are actually really complicated to do and involve lots of different systems in our body all working together in just the right way. How hard is it to actually stand up? And just think through all the different things that have to work in sync for you to be able to stand up easily from the chair
without using your hands. Yeah.
So these become biomarkers that geriatricians and other doctors use in clinical practice to measure the state of people's aging and the degree of their frailty. So literally, one test is called the timed up and go test. It measures how much time it takes for you to stand up from the chair, walk a few meters, turn around, and walk back. And that number is actually very, very predictive of, for example, how likely you are to have major complications after a surgery.
So what is that number or speed? No, because I'm always thinking, because I
See how
can say,
fast you can do it.
no, I'm thinking, okay, so can I ask my mother or my in-laws
Yeah.
to see?
Yeah.
I mean, for each of these tests, there'll be published reference ranges
Yeah,
that the two of us don't,
just
the two of us don't have on the tips of our fingers. But there are reference ranges for all these things, including things like hand grip strength, which are all like very easy to do. 4-meter gait speed, 6-minute walk or 400-meter walk. You could take your mother to a track and have her walk around. Like all of these things have very well characterized reference ranges that that are obviously going to be also age, sex specific. So you'd need to know where you fell in those brackets to give you an accurate number. But they're free to do, right?
Yeah.
And actually, over in France, we have some friends and collaborators in Toulouse, Bruno Vallis's group, and they're doing these remote ways of monitoring physical function. The program's called ICOPE, and they measure a range of different things around sensory function, but also physical function. And there's so many ways that we could do a better job of measuring how are you aging? And what their program does is try and raise flags if you move from being in the like, I'm good category to not so good. And then can we get in there early and intervene for those things? So many of these tests are things that can be assessed remotely without needing to bring you in and put you in an MRI scanner or hook you up to any IVs or anything like that. So I think they should be implemented more widely to track healthy aging.
The geriatricians like to say the most important thing you can learn about your patient's health that just totally changes the way that you think about them is just walking with them from the waiting room to your exam room and watching them walk. And the amount of information encoded in watching how someone's walk is just immense.
So that's the best measure of aging. So I always hear about aging clocks and other stuff, and then I always think, well, the geriatrician can fairly figure out within 10 minutes roughly how good or bad the state of the health of the patient is.
Yeah, nothing beats function.
Nothing beats function. So that's why physical activity is so important. So going back to this takeoff, we said that they have to be pre-filled, so that means they might also have existing conditions, because it's really hard to find 70 plus, 65 plus years old who are healthy.
Yep.
And nor would we want to for takeoff, because that wouldn't be very representative. That's a very small group, and that's not what most older people are like. So yeah, people will have various chronic diseases. They'll be taking various medications. So were the people in our pilot, even though they were pretty healthy in general.
We only had a short list of medications and conditions that we excluded. And we like people when they start to be in stable health. So we don't like someone who has any new diagnosis where they might be working with their doctor to change their medications or, have recently been in the hospital. So, it's sort of trying to strike that balance between pragmatism, but also scientifically, we don't want people who are likely to have a change in health status during the study period.
This is an important principle for doing geroscience related, or even just doing clinical studies of any disease of aging. Like the reality is most diseases of aging happen in people who have other diseases of aging too. That's normal. That is the normal biology. So if you really want to understand or study something like frailty, that doesn't happen in people with no chronic diseases. It happens in people who have, and those other diseases contribute to the biology. So if you want to really understand the condition, you have to study it in its natural state. Which is in the full complexity of the people expressing it.
And also it's very important that people who are going to sign up, and if you're listening to this, if you're watching it, then please search for his clinical trial. Takeoff is very easy to remember and search for. And if you ever want to do any personal contribution to medical research, I think enrolling in a clinical trial is the best gift you can give to humankind.
Yeah.
Because these trials are extremely difficult to carry out. Many years of planning, pre-planning goes into it, and a lot of honest taxpayers' money go into these trials. And these trials, numbering say 160 in this case, will produce results that will change the clinical practice, may change clinical practice at one day, and may affect 160 million people at least. So your own participation can actually change 1 million people. And this is really, when I started to think about it, and when we did the first firefighter study, there were only 150 firefighters. And then they said, well, if it is going to benefit our brothers and sisters firefighters, then we'll do it. And they were very diligent about it. They realized that participating in the trial, each one of them, is carrying the burden of health of 100,000 firefighters for years to come. So this is extremely important. So that's why I always ask people, hey, do I qualify?
Yeah,
Study,
absolutely.
because, this will be the best gift you can give to humankind.
Yeah.
Yeah, we've one of the, I mean, there's been a lot of satisfying things about initiating a clinical research program at the Buck. For both of us, I think it's been something that you wanted to do ever since you joined the Buck. But the day-to-day interactions that you get with the participants, everyone has their own reason for being there. All of the time, it's a completely selfless reason. They want to be there because of an aunt or a brother or some family member, or because they want to advance science. And it's nothing like the interaction you have with someone in a doctor's office where they kind of have to be there and they're worried and stressed, and all of the interactions are so positive that I think that it's just a very giving interaction between both research subjects and scientists. They have opportunities to ask questions, to learn more, to to find out things that they can implement to help themselves age better. And it's I obviously I'm speaking from the scientist's perspective, but we've had participants come back and want to be in other trials, people who have kept in touch and written us Christmas cards and things like that. So I think that if you join a trial, you plug into this network of contributing to science and learning and engaging with science that's got a lot of levels of reward to it for everyone involved.
Yeah, and also Buck Institute, I think is the only fully dedicated institute for aging research.
It is. It was the first institute solely dedicated to aging research.
25 years this year.
25 years ago.
Yep.
And now does the whole spectrum of aging research, from the most basic biochemistry and model organisms, worms and flies and yeast and all of that, all the way up through testing these ideas in clinical trials and clinical research now.
So both of you are also involved in educating and translating, or you can say spreading the word about aging research and geroscience. Can you tell us a little?
Yeah, geroscience is still such a small field. And I think everyone who's listening to this is probably thinking how impactful it might be and how important it is. But it's a very small field. And that's why it's so important to us to create this clinical research program to bring these ideas out of the laboratory and get them towards clinical research and clinical practice. But we're only two people, and there's not too many people like us doing these sorts of clinical trials. So one of the most important things to me to advance the whole field is we need more investigators who can do these early stage human biology proof of concept kind of clinical trials and clinical research studies. And so a big part of my career is being involved in education and training programs to tell medical students and PhD students that, hey, aging biology is a thing. Geroscience is a field. It's a real scientific enterprise that's moving towards the clinic. And also, hey, it's a fun field to do science. And if you're interested, we've created curricula and programs that you can get the training you need to learn about aging biology, to learn about doing translational research, learn about doing research with older adults, and so to grow our field and actually have more investigators who can do these studies. And hopefully, we've already watched this field grow from very, very small 10 years ago to
You're being generous, including me in that.
to much bigger now. And I think the field is going to grow exponentially in the coming 10 or 20 years.
Yeah, and also we are aging faster. So in 10 or 20 years, there'll be more older people. And in fact, people always think that, okay, so geroscience is going to keep a small group of rich people healthy. But actually, if we think about it, we want to keep people who are 60 to 65-year-old healthy and strong enough so that they can retire in a very healthy state and can enjoy life for at least 10, if not 20 more years.
Yeah.
So that's the goal. It's actually the goal is to keep everybody healthy beyond their retirement age, or even at the age of retirement, so that they can they can reap the benefit of a long working life that they have worked for 30 years to enjoy it.
Yeah, aging affects all of us.
Yeah.
And the founding mission of the National Institute on Aging, what you just said, Satchin, is that every older adult in the United States deserves the opportunity to enjoy the fruits of the life that they built as they age. That was the founding mission of the NIA 50 years ago. And I love that message. And I love that aging is universal, that it affects us all, and geroscience, trying to intervene on the parts of aging that are bad for our health, so we can live healthier longer, will benefit all of us, literally all of us.
Yeah.
It's so exciting.
Anything?
Oh no, I mean that's a great way to finish. I think it's an exciting field, and I feel like coming in, I feel very fortunate to have found it relatively earlier in my career, and I think that there's an awful lot of energy in the space, an awful lot of smart people in the space, and just feel very fortunate to be kind of like riding the wave, and hopefully it's transformative in the future for everyone.
Well, I think in general, almost all, not all, biomedical research, those who are doing biomedical research, for example, I started with circadian rhythm, and then of course, I have studied enough young mice and young fruit flies, and what I realized is that the older mice are very different. They even don't eat and fast regularly, and then they don't even have sleep regularly. So I guess all biomedical research will ultimately move towards understanding how the stuff that you are working on, whether it's cell division or whether it's energy homeostasis, metabolism, or immune function, what happens to that system when the mouse is old or when we are old. So I guess all biomedical research, the roars go to.
It'd be interesting, right? Because in the last, I feel like we've witnessed the shift from where, all mass experiments used to be done in like male black sixes, and now it's a requirement that we do both sex-specific male and female mice, and maybe even multiple strains, right? I wonder how long it'll be before it's mandated that we do both young and old cohorts in like all experiments, because then truly everything becomes an experiment that involves aging.
Yeah. Okay.
They have to make bigger grants before they make everyone do that, right?
Well, that's not good.
Yeah.
Thank you so much, John, and thank you, Brianna. And
Thank you.
My pleasure.
It's really exciting. We could have gone for another 15 hours or so.
Yeah, I think so.
I guess we will.
Maybe we'll start burning some ketones before.
Yeah, I guess maybe next time we'll have we'll have some ketone meters, and we'll measure ketone and talk until our ketone goes up to 0.2.
Sure.
Thank you so much, and have a wonderful day.
Thank you.
Thank you.